Okay, thank you very much. Thank the organizers for inviting me for the talk today. Okay, so I'm going to talk about single-cell transcriptomics, and generally like to start. I mean, Mikhail's actually already introduced this very well, so I don't have to spend very much time on the first few slides. Essentially, eukaryotic transcription occurs in pulses, and the consequence of that is you get large variation, which means that single-cell gene expression data is intrinsically noisy. Now, this is pretty much accepted now, but there's always arguments about whether bursting affects all genes or not. So I just put in this sort of reference from 2012, where they actually assayed looking at about 8,000 genomic loci in the human genome. And basically it is that transcriptional bursting is the norm, it's not the exception. And then we already saw this from Mikhail, so again I don't have to spend much time. But essentially when you look on a linear scale, you see the effects of bursting that there are a few cells with a large number of transcripts, but most cells have a much smaller number. And so that the average that you get in most, in typical population experiments is really dominated by a few cells that have a large number of transcripts. Okay. So what does this mean practically? So how do you address this noise that's an inherent part of looking at single-cell transcriptomics? And basically what you need to do is you need to look at multiple cells and many genes. Okay? And this is, again, so how statisticians want to express noise is by collecting more data. And so sort of Fluidigm got started with single cells with these dynamic arrays that again were introduced by Mikhail. So this is the 96.96 array, and these are analyzed in the Biomark HD instrument. So on the 96.96 array, you put 96 assays pipetted into this side, and 96 samples pipetted in this side, and then under air pressure, these are driven into a PDMS chip in the middle of the array, so that you get all pairwise combinations of each assay with each sample. And so single-cell researchers started using this because they could then look at 96 different transcripts in 96 individual cells. Okay. And so we actually published a detailed protocol for how to do the dynamic array analysis on FACS-sorted cells. And then this protocol was then used in the following study, which I'll spend some time talking about. Okay, and this is actually looking at genotype-phenotype correlation at the single-cell level. And this study was largely driven by Quinn Wills, who was at Oxford at the time and is now back at Oxford. And again, it's sort of looking at what power you gain by having single-cell resolution when you're trying to look at genotype-phenotype correlations. Okay, so this is how the study was set up. Basically, we used lymphoid cell lines from 15 of the HapMap samples. So that's how we get the genotypes for these individual cells, because these were genotyped as part of the HapMap project. Okay, and then to have some sort of biological effect, we treated with a Wnt pathway agonist. And then we analyzed those cells using 96 different qPCR assays. So we could look at 96 different transcripts. So here we had 15 cell lines. And for each cell line, there are two conditions. There's the unperturbed condition and then perturbed with the Wnt agonist. For each of these conditions, we used 48 single cells. So that's actually a total of 1,440 samples, and for each one, 96 different assays. So a little bit over 138,000 data points, qPCR data points. And so because we were using the 96 arrays, we were able to analyze that on 15 arrays and could do the analysis, the qPCR part of the analysis, in about 2 weeks. If this were done in 3D 4-well plates, it would have taken 384 well plates. So this parallel processing that you can achieve using the dynamic array, this really illustrates why it's useful to have that parallel processing. Okay. So first I just wanted to start with a control experiment that we did. So these are actually naive B cells isolated from 3 unrelated donors. Okay, so these are from 3 different human individuals. And then what's shown here is for the 49 genes that were detected in at least 50% of the cells. These are essentially represent box plots. Okay, so the 25th to 75th quartile, and there are 3 bars here for each of the 3 different individuals. Okay, and the main thing you can take away from this is that, in general, the variation, the cell-to-cell variation within an individual is greater than it's much greater than the variation between individuals. Okay? This really illustrates the kind of heterogeneity you see at the single cell level. Now there are other interesting aspects of this data, so that any particular gene has its own characteristic distribution. So here you can see June up here that has a very narrow distribution, whereas PRKCE has a very sort of broad distribution. So in all of these genes, there's actually only 2 genes that actually show variation among the different individuals that gets to be on the order of what you see as a cell-to-cell variation between individual cells. Now then, one other aspect of this data that you can sort of see here is that there are really sort of 2 aspects to looking at single-cell data. Okay, so over here you sort of see what I call continuous data. Okay, where you get the sort of normal distributions that Mikhail was showing distribution plots of. But then what's shown over in here in parentheses, this actually represents the % of cells where you did not detect the transcript. So up here for beta-actin and GAPDH, those were actually detected in all the cells analyzed, whereas here for cremin, it was detected in about 50% of the cells and not detected in about 50% of the cells. So in addition to sort of this continuous aspect of the data, you also have this sort of discrete on-off data. And I know in some ways this sort of reminds me like the duality of light, right? You can think about light as a wave or as a particle. Looking at single-cell transcript data, it has this sort of continuous wave-like characteristic, but it also has this sort of discrete particle-like characteristic. It's actually important to take into account these 2 different aspects of the data when you sort of do the analysis. Okay, so then so why do this on single cells? Well, if you just use bulk measurements, you'd have 96 different genes, but the only phenotype you would have is you'd have the mean expression level for each of those 96 genes over all the cells in each group. So, you essentially only have 96 parameters to analyze genotype-phenotype correlations. By having single-cell resolution, you can actually add all of these different phenotypes to looking at what correlations are occurring between the genotype of the 15 different cell lines and its response to the Wnt agonist. Okay, and so you have things here like expression noise. So, how broad is that distribution you're looking at? You can From the data, you can estimate burst size and burst frequency. And then here, as Mikhail again pointed out, it's really important to look at correlations. So here are 2 phenotypes that are really based on looking at correlations of having the same two genes expressed in the same cell at the same time. And then here are some other things looking at, we actually had markers for looking at whether you were in G1 phase or S phase. And also the number of cells where you did not detect the transcript was used as one of the phenotypes. And so again, Quinn sort of addressed this sort of continuous versus discrete aspects of the data by having different phenotypes. These, when you look at correlations, he used only the continuous aspect of the data. Okay, and then these final 2 phenotypes, here he used only whether it was on or off, so the sort of digital, discrete aspects of the data. So in this study, and then these other 3 actually have a sort of a mixture of using both the continuous and the on-off data. So by having these different phenotypes, you can actually highlight different aspects of the data and see if you get significant correlations by looking at these different aspects in continuous versus discrete data. Okay, so then why is it important to do this? So basically we were able to identify 47 significant associations by using the single cell phenotypes. And this was even by increasing the stringency of the multiple testing threshold by a factor of 10. Okay. Using just the average data, there were only 6 significant associations that were found. And again, this is using the more normal sort of multiple testing threshold that human geneticists use. Okay. So basically, 8 times more associations found by having single cell resolution compared to using just the mean expression data. And 7 of the genes that had these significant associations had actually been implicated in clinical GWAS studies, but previous studies had not been able to find any EQTLs associated with these 7 genes. So in this case, Quinn likes to call these SCQTLs, or single cell EQTLs. Okay. So then what sort of lessons can we learn here from this particular study using qPCR to look at single cells? The main point is single cell data increases the number of phenotypes, and so it increases the power of finding associations linked SNPs to function. Okay. In this particular case, people always ask us, 48 cells per condition was enough to get statistically significant results. And then finally, the one thing I wanted to emphasize is you really need, in single cell data, need to look at both the continuous and discrete aspects of the data. And you need to explicitly distinguish those and test those in looking for associations. And I think this is early. I think it's too early for there to be a consensus on how best to do this. So Quinn had one particular way of doing it. I know of at least 2 other publications that use slightly different ways. But again, they look explicitly at continuous and discrete aspects of the data. Okay, so with the use of the biomark for doing single cell studies, Fluidigm decided to invest on a product sort of really dedicated for single cell analysis. And that's where he came up with this C1 auto prep system. So I want to emphasize that this is a sample prep system. Okay, so the biomark and using the dynamic arrays is an analytical system that you do qPCR in. This is a sample prep system that lets you isolate single cells and then do a series of biochemical steps. And then with the libraries you create from the single cells, you can do a number of different analyses. So it is possible to use the C1 to do qPCR like I just talked about. But what I want to talk about is using the C1 for doing RNA-seq. Okay, so mRNA-seq from single cells. So basically, within the chip in the C1, you flow through a suspension of cells, and it captures individual cells, up to 96 individual cells in a capture structure within the chip. And then once they're captured, you can do a series of up to 5 biochemical steps to process those cells to get various kinds of libraries. For doing mRNA-seq, within the chip, you actually do the smarter chemistry from CloneTech. And this is a template switching mechanism to generate cDNA libraries. And then these cDNA libraries are harvested from the chip. And when you take those harvested libraries and then use the Nextera technology from Illumina to fragment the cDNA libraries and to put on the sequencing adapters and then do sequencing. Okay, and so this represents a collaboration that's been ongoing between Aviv Regev's lab at the Broad Institute and Fluidigm. Rahul Satya is actually here at the meeting. He'll be talking tomorrow. So if you have questions about this, you can ask him about that. So I'm actually not going to talk anything about the biology here. I really want to talk about 2 of the control analyses that Rahul and his colleagues did. And also I should mention this study will be published in the next few weeks. This study will actually be published. Okay, so the control experiments I want to look at doing RNA-seq. First of all, this just really graphically shows you the cell-to-cell heterogeneity, now looking at all the transcripts you can detect by RNA-seq rather than just 96 at a time. And so basically when you compare one single cell to another single cell for this particular type of cell, the correlation between the two cells is only 0.57 correlation coefficient. Okay, now when you average together about 100 single cells together, you now approach the levels and you get good correlation with the bulk data. So again, this shows graphically how much variation there is from one cell to the other. Now this high degree of variation actually has an interesting consequence, because in a practical sense, what it means is you don't actually have to sequence that deep when you're doing single-cell transcriptomics. Okay, so this now goes another different control experiment where you just take a library, this could be any RNA-seq library, and you compare a MySeq run to a HiSeq run. So essentially, the MySeq run shows the noise you get by reading too shallow. So this here, and then there's these where you see zeros along here, that's the noise you get by having a too shallow read on your RNA-seq library. Okay? But you can see that this noise is really dwarfed by the cell-to-cell variation. Okay? So this, right? So the variability in sampling due to shallow read depth is really insignificant compared with the biological variability you get from cell to cell. And so what this means in the practical sense is, I mean, if you keep reading deeper, you're getting more information, but you're really better off spending your money looking at more single cells at a shallow depth than you are by looking at a few cells at high depth. Okay? And again, and that we heard earlier in the talk about this paper from Ito Amit in Science. In some cases, they only actually did 20,000 reads per single cell, and we're still able to get data that was useful for them. In this particular study from the Broad, they ended up using on the order of 1 million to 2 million reads per single cell. Okay, then I want to close by talking about this paper that was just recently published. So this study sort of addresses the noise due to asynchrony in single cells. And this is largely the work of Cole Trapnell and Davide Cacciarelli. Okay, and so Cole was mainly responsible for the sort of mathematics I'll be talking about, and Davide was the driving person looking at the developmental biology. Okay, and so these are individuals from Harvard University and from the Broad. Okay, so what Cole came up with is this sort of new algorithm that he calls Monocle. And basically it's for analyzing single-cell RNA-seq data. And it orders cells by progress through a developmental process. And then you can classify cells according to type, and then find differentially expressed genes, and look at clusters of genes according to temporal patterns of expression. And the really important thing is that Monocle is completely unsupervised. Okay, so that it can do these things without knowing which genes are important ahead of time. The developmental process they looked at was looking at a myoblast differentiation. So they started with primary human myoblasts. And then this is a process that's been studied for at least 30 years. And so by switching to low serum, you can induce these myoblasts to differentiate. Eventually, they'll actually generate twitching cardiac muscle cells. Okay, and then this shows what they did was they did 4 different time points. So 0 time point, and then 24, 48, and 72 hours. This shows that this was, again, done on the C1 instrument, so you can capture up to 96 cells. This actually shows the number of cells captured in each of the C1 runs. and then this is the reads per cell in terms of millions of reads per cell. So they went to the depth of like 4 to 5 million reads. You notice over here that sort of the capture rate sort of went down here. That's because you're actually, at 72 hours, you're actually starting to get fusion of cells. So you're starting to get some larger cells and things in there. Okay, and this basically shows the problem you get when you do this type of experiment, in that myoblast differentiation is unsynchronized when you do that experiment. Okay, so these show the different time points, and so you have 0, 24, and 72 hours. But even at 0 time point, this is this is a gene that should be expressed in the differentiated cells. So you should see this at the 72-hour time point. But even at the 0 hour time point, there's still a few cells that actually show expression of MYH3. So I imagine we have like at least a few professors and things. It's like any sort of class you have. There are sort of a few students that sort of zoom ahead and get there really quickly, and some others that sort of lag behind and sort of stay in the rear. And the same thing happens when you try to do these sort of in vitro differentiation experiments. And then this just shows another example, ENO3, again showing that this again should be a late marker, but you actually see when you look at the average measurements for ENO3 across the different time points, you actually don't see much evidence for the thing actually increasing with time. Okay, so then this outlines the algorithm, the monocle algorithm. So basically the first thing you do is you take each cell and you represent it as a vector in multidimensional space. Okay, this is mRNA-seq data. So that means the number of dimensions is on the order of 10,000 because you detect on the order of 10,000 genes in a single cell. Okay, so you have each cell here in like 10,000 space. And so to deal with that, you have to sort of reduce the dimensions. And so what was used here is something called independent component analysis. So independent component analysis is like a cousin of principal component analysis. So computer scientists actually use this to study something they call the cocktail party problem. So if people are interested in discussing the cocktail party problem, I think that's really appropriate for the wine session, so we can do that later. So in this case, they've reduced down from like 10,000 dimensions down to 2 dimensions. And then once you have this in 2 dimensions, then you construct a minimum spanning tree, and then you can order the cells along this minimum spanning tree, and you can also identify, based on the genes expressed in these different cells, you can identify the cells by type. Okay, so I'll just go step by step through this process. So this shows all the cells in 2 dimensions after doing the independent component analysis. And then the first thing you do is construct the minimum spanning tree, and then you find the longest path through that minimum spanning tree. And then again, once you have these cells displayed like this, you can start using the actual genes that were you actually see detected in these various cells to identify the different cell types. So in this case, they were able to identify proliferating cells. So these are actually early in the process, and then differentiating cells. Okay, and then also there were contaminating fibroblasts that were identified. So we heard in the first talk this morning that we have to be really careful about purifying cells. This suggests that when you have one reason for getting single cell resolution is that maybe you don't have to be so careful. If you can identify those cells informatically, then you can eliminate them from analysis using bioinformatics rather than having to do purification. Okay? And then finally what's constructed is what you call like a pseudo-time axis. And so you know that proliferating cells should come before differentiating cells. So the pseudo-time axis starts here at time 0 and then goes through pseudo-time 0. So now you can look at the cells along the pseudo-time axis rather than the real-time axis that you did when you collected the data. So this shows ENO3 where you now look along this pseudo-time axis. Okay, and so what you can see is that you can see really strikingly here that here are these yellow cells, which were actually 24-hour cells. Some of them have now moved out to the end of the process. So in 24 hours, they've actually made it as far, they've made it all the way to the end, and some cells take 72 hours. And there are a few 72 hours here, cells that never even got started. They just sort of stayed back at the beginning. And this is just to remind you what this looked like when you actually tried to do this by the time, 0, 24, 48, and 72. And so now you can clearly see that ENO3 is a gene that's induced as part of as a differentiation process. And this just gives some more examples. So CDK1 is known to be a proliferating cell marker, and so when you now look at it under pseudotime, you can really see that it's being turned off during the process of differentiation. And this is like a really interesting case here, these next two genes. So it had been inferred from mouse studies that as you decrease the expression of ID1, you should be inducing the expression of MyoG. And if you look at this average data, it's hard really to make that conclusion that ID1 is actually loss of ID1 is driving induction of MyoG. Whereas when you look on pseudotime, it now becomes very clear that as you see ID1 decrease in expression, you now see the increase in MyoG expression. And then what you can do now again is order the cells along the pseudotime axis and look for differentially expressed genes. And so when you do that, you can now actually identify over 1,000 genes that are dynamically regulated using a false discovery rate of 5%. Okay, and so this now identifies genes involved in the developmental process where you're actually analyzing it along the developmental process, not your experimental time points. And finally, I wanted to say doing this analysis just didn't tell us things that were already known. They could actually use this analysis to identify new transcription factors that had not been discovered before. And again, I said people have been studying this for over 30 years, and so this was still able to find 8 transcription factors, right, that inhibit muscle differentiation that were previously unknown. So I don't have the time to go through this in detail, but basically it is by having that pseudo-time data that I showed you on that last slide, they could cluster genes into 6 distinct trends of up and down regulation. And then what they did was they looked at each of these clusters and they looked for cis regulatory elements that were in common within any given cluster. And this identified candidate transcriptional factors that they then could look at. And then these candidates were then analyzed using shRNA knockdown experiments. And that's how these 8 factors were verified as factors that actually inhibit muscle differentiation. Okay, so with that, I will go ahead and close. And so I really sort of think the unifying topic that sort of connects what I was talking about is we're sort of really looking at noise and single cell transcription, right? And there are various types of noise. And the one is the intrinsic noise due to bursting. And that's sort of the predominant noise you get when you look at the single cell data. And again, as we heard this morning, it's an open question of how much of this noise is nuisance and how much of it is actually integral to the way biology works. And so that's what we're all interested in studying. Now, there is technical noise, and you should always work to sort of minimize technical noise. But the good news, in a way, is that this technical noise is generally not limited in doing single-cell studies. There's such a large amount of biological noise that, again, you should be as careful as you can, but it's not going to limit the sort of results you get. And I also there's also, when you're doing in vitro experiments especially, asynchrony noise and looking at developmental processes. And so how do you address that noise? The first thing is to look at many cells and many genes. Of course, mRNA-seq lets you look at a large number of genes. In these 2 cases, a few hundred cells was enough to get significant results. These are relatively These are relatively simple experimental systems. In the paper that's going to come out from the Regev lab, that's a much more complex process looking at the immune dendritic cells. And there they ended up looking at a few thousand cells. It's important to explicitly address continuous and discrete aspects of the data. And then I told you a little bit about Monocle, which is one bioinformatic way of trying to address the asynchrony noise. And I'm sure over the time there will be other bioinformatic tools that will help people address the noise in single-cell data. So with that, I'd like to thank you and hope we have time for a couple of questions.
Very interesting. I really like that monocle. That looks like it's going to be very powerful. My question is about set point and homeostasis. So if you have a distribution of gene expression for resting basal cells, then you perturb those cells, stimulating or suppressing. Do they go back to their same baseline in the distribution, or do they go to a new set point? And how do they know what set point they should go back to? We ask this question all the time with proteomics. If you stimulate a cell, giving it, with an EGF, and you turn on the receptor and all the downstream activities, then they go back. Somehow they all go back to the same set point afterwards. I'm asking the same question about what your opinion is. This data, of course, doesn't answer that. What's your opinion about whether they go back to the same set point specific for that individual cell, or whether it then randomly gets set to a new set point?
I haven't really thought about things that way. I mean, the one thing that does seem, it's not quite answering your question, but one thing that does seem sort of clear from looking at these single-cell data is that when you actually do induce something, what is happening during that induction process, as you see, if you're looking at average data and you see it rising, what is happening is not that every cell is increasing by a little bit. It's that you're recruiting more and more single cells to start generating that. So, certainly the induced state is a new level. But then when you take away the induction, I guess I'm in the opinion I'd probably go back to the same sort of stasis point. I don't know, Mikhail might have thought about this as well. No? Okay. We can talk about that over wine as well.
All right. Yeah. Sorry, is there a question? Oh, yeah.
No, you stated that technical noise is generally not limiting in single-cell studies, which is a bit surprising to me. What would you do if you have two methods to get your data, your technology, let's say transcriptomic data, and you would like to differentiate between bursting and technical noise? How do you find out what is the contribution of each aspect?
Yeah, again, I don't know if I can answer that right off. Now, it's actually very interesting in looking at single-cell RNA expression. We actually have two completely different ways of looking at that data. So one is sort of like by the molecular biology things that we were talking about here, but then the other is by doing in situ hybridization, which Mikhail referred to a little bit. And Raul actually has some very interesting data he was telling me about in terms of looking at the comparison, looking at in situ hybridization versus RNA-seq data. And that overall, you can see a correlation between these two different ways of doing it. That seems like to be the only way you actually really can look at what is the technical noise for any one given technique, is by having something, a sort of orthogonal way of looking at the same sort of data. Mikhail?
Well, I can comment on one thing on that. Typically, if you have protocol and you want to distinguish the noise contributions from different levels, you can always do a nested design on that. And we have actually done and published that on single cells. So essentially, collecting single cells, you take the single cell, you lyse it, and you lyse it in replicates, you do RT in replicates, and you do qPCR in replicates, and then you can calculate the standard deviation from the different contributions. Typically, the standard deviations among subjects, it depends a lot on the gene you're studying, and it can be anything from 0.5 cycles to 2 cycles. On the single cell level, it varies also, but the variation across individual cells is actually a standard deviation of about 2 cycles. There's a 4-fold variation. Ken has even a plot that shows it varies also across genes and cells, but it's very, very large. The standard deviation of the RT is typically of the order of 0.3, and the standard deviation of the qPCR is of the order of 0.1. So there are very, very large differences here, and by far the largest contribution is the variation across the single cells. Anyhow, let's move on to the next speaker today.